Okay, so in the next hour and a half, we are going to hear from 6 speakers who are going to talk for 15 minutes each. I'm imploring them to stay as close to possible as on time. And the first speaker is Klaudia Kuranda. And Klaudia actually trained at the University of Toulouse and then postdoced in Paris and was a scientist at Salpêtrière until 2018 when she moved to Spark Therapeutics. So she's had a history in both academia and biotech and has worked to try to understand human immune responses to AAVs. So Klaudia will be talking about AAV-specific antibodies and how they modulate immune responses to AAV. Thank you.
Thank you for the introduction and thank you for the invitation. Okay. Does not work. Help. Okay. Should I? Oh man. Okay. Now we are in the middle of my presentation. I'm going to get this under control. Sorry. Please bear with me. Here we are. Okay. So I know we've went just through the very, exceptional and very comprehensive introduction. So I'm just going to mention that, of course, the systemic AAV administration is used very often, mainly for hematologic, metabolic, and neuromuscular disorders. So with this route of administration, AAV is infused directly into the bloodstream where it's very susceptible to the neutralizing antibodies. And those pre-existing antibody, their prevalence differs depending on the serotype. You can see an example here. They may differ with geographical location. And overall, they increase with age. So now, to solve the problem of pre-existing antibodies and how they limit the access of the seropositive patients to the gene therapies, of course, many groups work on the antibody removal techniques, and they seem to be very efficient. And as we have seen, they can enable transduction in previously seropositive animals, for example. And however, they cannot remove antibodies completely, right? They lower them. And also, they do not really address those pre-existing memory cells, right, that can be present in seropositive individuals. So my question is really, how would look like the immune response in a patient who received, who was seropositive, his antibodies, bulk of the antibodies was removed, and now he's receiving a gene transfer, right? Would the cells that are their memory cells and the remaining levels of antibody enhance the immune response, or maybe not at all? So just a reminder, of course, antibodies mediate many aspects of antiviral immune response, like the neutralization, opsonization that will enhance viral clearance, and complement activation, very important part. So complement activation together with antibodies will facilitate even more opsonization, and importantly will drive the inflammation in response to a virus. And how about AAV? AAV, of course, is not an exception here. Importantly, cases of complement activation in response to AAV have been shown in clinical trials using high doses of AAV. They were linked to the cases of thrombotic microangiopathy, which can be actually quite serious and is a threat to the safety of the patients. Some patients needed to be hospitalized. So we were trying to answer this question of how antibodies may affect immune responses using the human blood. And you can see here we stratified them, we divided them on groups depending on their content of neutralizing antibodies. And we could show that actually in vitro, the presence of those antibodies in blood will enhance pro-inflammatory cytokine release in those samples. Earlier, Zeiss and colleagues have shown that AAV can interact with complements. You can see it here, and this activation, so activation of the complement pathway is prevented by EGTA. EGTA can block classical complement pathway that is dependent on antibodies. So they have suggested that actually complement activation in response to AAV is antibody dependent. And we could show this later, again, using blood samples, where you can see sample without antibodies and increasing titers of antibodies, the complement activation will be much higher. We could also show this by mixing plasma samples, NAP positive and NAP negative plasma samples in different proportions. So you can see here that there is a direct dependence of antibody titers and complement activation. Later, other groups have shown very nicely that when we deplete IgG antibodies, we can also prevent complement activation in response to AAV, and that was done two different ways. So by depleting antibodies through immunoprecipitation or cleaving them with IDS enzyme. And also another group have shown the same data, just that additionally, they shown that it's much more efficient when we cleave both IgG and IgM to prevent complement activation than when we cleave only IgG or only IgM. So this data, though, they are done, of course, ex vivo, right, using human blood, but they show us that actually antibodies can enhance complement activation and then enhance release of cytokines in response to AAV. But what about the cellular component of the immune response? So here you can see, again, blood samples with different titers of antibodies and how they can enhance the uptake of AAV particles. Okay, so you can see monocytes and neutrophils here that will, of course, phagocytose, and they will mediate inflammation as well, right? But also the dendritic cells that can present AAV antigen and prime later T cells. And another study have shown that in seronegative healthy individuals, in contrast to the seropositive individuals, we can detect CD8 memory cells that are AAV specific. Of course, the B cells, antibody secreting cells, right, that produce those AAV antibodies. But they do not contain NK cells that are reactive to AAV that would secrete the interferon gamma. So basically, you see that even the cellular components of immune system are changed upon this natural infection with wild-type AAV. But of course, the most important data comes from the clinic, right? So here you can see the hemophilia B therapy trial that included NAP positive and NAP negative patients with rather, let's say, low neutralizing titers. And you can see that even the effect on the transgene expression here of those antibodies it was there, but it was low. And definitely all those patients could benefit from the treatment. Interestingly, both groups of patients had liver enzyme elevations. I mean, some cases, right? Not all of them, some cases. However, the NAP positive patients, the time from infusion to the first ALT elevation was faster than in the NAP negative patients. So it was 4 weeks instead of above 7 weeks on average. And the duration of ALT elevation was also longer in NAP-positive patients. So it was 50 days on average versus 33 days. Similar study was done in NHPs. Here I'm not going to show this data due to the time restriction here. But it showed similar findings that basically there was a low impact on the transduction, but the transduction could be achieved in some animals. But basically, there was none of the studies shown enhanced immune response in the NAP-positive animals or patients. So what I think is important, both of those studies used AAV5 serotype. And it has been shown that antibodies, neutralizing antibodies that recognize AAV5, they have very low affinity compared to antibodies that can neutralize AAV2 or AAV8. So basically, it's possible that the neutralizing assay that we use in vitro will show you that those samples are positive. However, in vitro, as those antibodies have lower affinity, they may not be so efficient. That could explain why they have such a lower impact at the neutralization. But as we have seen this morning, there is some evidence also with other serotypes. So another hemophilia B trial done with AAV2. And here we have this patient that we saw this morning. The NAP titer was only 1 to 2 compared to the patient with higher NAP titer, 1 to 17. Here there was a strong effect on the transduction of the transgene. But again, here we didn't see liver toxicity why we could see it here. So here, of course, the caveat are that it's N of 1, right? So it's very low. It's hard to have maybe a very strong conclusions here. And also the dose of the vector was very low. So basically that's below the threshold of, for example, complement activation, right? So it's, I think there are all those questions that remain here. So just to conclude, I think that for me, at least personally, it's not really clear what would be the immune response in AAV seropositive person, because this evidence, I think so far, seems to be limited to AAV5, right? The evidence of the safety. However, the pre-existing sorry. Oh no, it's gone now. Okay, well, I'll do it without the slide. It's coming back. Sorry. I'm like, up. No, we are here. And I cannot do it full screen here, but okay. So basically the antibodies, when we measure them in the blood, but ex vivo, right, they can enhance inflammation because they increase complement activation, because they increase cytokine release. They will increase antigen uptake into the dendritic cells. On the other hand, they could also decrease inflammation because they prevent transduction. So AAV will not be in the targeted cells, right? So you will erase all this potential innate sensing in the cell, like DNA sensing, right? RNA sensing. And you will enhance vector clearance. So basically, you kind of will remove those antibodies from circulation before they have a chance to give maybe a big pro-inflammatory response or some immune-related toxicities. And the point that Shari mentioned as well, that antibody removal techniques, they remove majority bulk of antibodies, but they do not take care of the cellular immune responses, right? That will be primed, those AAV-specific T cells, ready to respond maybe faster than in just seronegative patient. And yes, and I think that maybe more studies would be needed to using other AAV serotypes than AAV5 to confirm that this is safe, and maybe that we can use the standard immunosuppressive regimens, because I think that for the moment, even in seronegative patients, sometimes steroids, right, lack efficacy. In some cases, very rare cases, but it happens. We don't control them completely, or they do not prevent TMA, for example, right? So what can we expect in seropositive population? Shall we have some specific immunosuppressive regimens that could address maybe those immune responses? Well, so that's a lot of questions. Thank you so much. Sorry for the technical problems here, and I would like to thank all my collaborators and friends that work with me. Thank you.
Okay, great. And actually, I failed to mention earlier that we are going to go through the 6 talks and then have a panel discussion, so save your questions, and we'll hear them anon. So our next speaker is Dr. Brian Long. Dr. Long trained in immunology at UNC Chapel Hill and postdoc at the Gladstone Institute, and has worked in several different industry settings. At BioMarin, he developed neutralizing antibody assays for AAV and actually described the phenomenon of non-antibody transduction inhibition in the course of doing that work. So we're looking forward to his talk, overview of AAV antibody assay development and efforts towards standardization.
There we go. Better? Okay, thank you very much for the invitation to speak today. So the purpose of my talk today is really to kind of go over some of these assay formats that we use to measure pre-existing immunity, or specifically pre-existing antibodies. And really what that takes the form of are assays that measure neutralizing antibodies, or the neutralization effect of a patient's plasma with pre-existing immunity, or binding antibodies to the capsid. I'm going to talk about some of the discrepancies in those assays, where they're concordant and whether they're discordant, and what the effect of that might be. And then I'm going to go through some case studies where these assays have been applied to pre-select patients for pivotal phase 3 clinical trials. So I always kind of like to start with this slide. Federico kind of already went over this in his previous talk as well. It just kind of sets the stage for what we're talking about when we're looking at immune responses to AAV vectors. So what's being shown here is basically a schematic of an AAV capsid, and inside of that is the transgene cassette that encodes that has the cDNA that encodes the therapeutic protein. When we're thinking about immune responses to AAV, we have to consider all of these components. Most prominently is this capsid coat protein that elicits the antibody response for the most part. But you can also have responses to the therapeutic protein that's being produced. And you can also have some of these innate responses that are more specific to the nucleotide sequence being held within that cDNA. Most prominently are unmethylated CpG motifs that activate TLR9. But mostly what I'm going to be talking about today is this antibody response to the capsid, and that's illustrated here. So for those of you that don't know, antibodies are basically produced by B cells. What's being shown here is an IgG isotype of antibody. This end here is called the FAB region that binds to the capsid. This is kind of the effector end of the molecule. At this end is the FC region that can interact with the cell surface of other effector cells. But really, I'm going to be focusing my talk today on how we actually measure these antibodies or neutralizing activity within the plasma of patients that might have pre-existing immunity. So I'm going to start with the traditional cell-based NAb assay, also sometimes called the transduction inhibition assay. Basically, what this assay is doing is measuring the neutralizing activity of whatever might be in a patient's plasma. What's being illustrated here are basically 2 patient samples. The one here on the left is a sample that doesn't have any pre-existing antibody to the AAV capsid. Over here on the right is one that does. You can see the antibodies that are bound to the capsid. Basically what we're doing is taking a reporter transgene. So if we just kind of go back to the previous slide, you can have a therapeutic protein here, or you can have one that encodes something like GFP or luciferase that then makes this a reporter when it transduces the cell. So we incubate these reporter capsids with the plasma. If there's antibody there on the right, it'll bind to the capsid. We then add it to a 96-well plate, and in that plate, on the plate surface, are a reporter cell line that is transducible by the AAV. So that's just being shown here, some cells on the plate surface. What happens next after the incubation is that in the absence of antibody, these capsids are able to transduce the cell line, bring the transgene DNA into the nucleus and express the protein. You can see here on the right that that's being inhibited by the antibodies. The capsids are not able to get into the cell. They're not able to transduce the nucleus. And so in the case where you do have good transduction, that leads to expression of the fluorescent protein and leads to the signal that you can detect in this cell line. Whereas on the right, because it's been blocked from getting into the cell, you don't have any signal coming through. So that's basically how this assay works. You can semi-quantitatively look at these things, measuring the titer of the antibody here. Whoops. It is a little bit of a touchy remote. It moves pretty quick when you're not paying attention to it. So here on the right is an example where there's antibody present, but as people have talked about, if you get excess capsids, the amount of capsid begins to exceed the neutralizing capacity of the antibody, and you might get some transduction, which leads to less signal versus the greater signals you see here with no antibody. In some cases, you can get multiple capsids getting into one cell, multiple genomes, much higher signal being expressed there. And that's how we quantitate the amount of antibody that is there by titrating that out. The next suite of assays I'm going to talk about are more ELISA-based assays. So here we're actually measuring antibodies that are binding to the capsid, whereas in the previous assay, we weren't directly measuring antibody. We were just looking at the neutralizing capacity of that plasmid. Here we're directly measuring the antibody, and there's a couple different ways you can do that. The first one here on the left is just an isotype-specific type of assay where you specifically measure either the IgG or the IgM antibodies that are binding to the capsid. You basically incubate a capsid on the plate surface and then add the plasma that has the antibody. Those antibodies will bind to the capsid. And then in order to detect that signal, you add a secondary antibody that has a substrate on it that is either fluorescent or colorimetric and changes the nature of the substrate so that you can see the signal. What's being shown here is, again, an isotype-specific type of assay where this detection antibody that you see here is specific for the FC region of IgG in this case, so the gamma chain or the FC region. So here we're just detecting IgG. You can use other assays where you're able to detect both IgG illustrated here and also IgM binding to the capsid, and one way to detect that is using anti-cap or lambda light chain detection antibodies. So instead of just detecting the FC region here and being specific, you're getting the light chains that are common to all of the isotypes of antibodies. So this is really more of a tab assay. Over here would be a binding antibody assay, and here a total binding antibody assay. Another way to measure a total binding antibody is to use a bridging ELISA assay. So again, for this, you coat the capsids onto the surface of the plate, add your plasma sample that has the antibody in it. Here, both IgG and IgM are able to bind to the capsid. And then you use a different type of reporter capsid. This time, it's not necessarily expressing GFP, but might be labeled with ruthenium. It's called a bridging assay because these antibodies are able to bridge the two capsids together, both the unlabeled one on the surface of the plate and the labeled capsid that's in the medium, and then this amount of binding is proportional to the signal that you get off the plate. And again, titers to these are measured just by diluting the plasma to the point where you no longer get a signal. Okay, so both of these antibodies, whether they're neutralizing antibodies or whether they're binding antibodies, can have an impact on the safety and efficacy of gene therapy administration. What's being shown here, and I think other people have shown a similar slide before, is that you can just have direct neutralization of the capsid. The antibody binds and prevents the capsid from getting into the cell, prevents binding to the receptor so you don't get transduction. Another thing that can happen is just opsonization of the capsid where the antibody binds. It may not necessarily neutralize, but it causes uptake into cells of the reticuloendothelial system, primarily just macrophages in the spleen, either through the complement receptor or FC receptors that are on these cells. And this causes like off-target a redistribution of the capsid where it's getting into the wrong tissue. There's been some evidence that shows it really goes to the spleen for the most part under these circumstances and not into the target tissue. So these assays, excuse me, getting over a bit of a cold, so my mouth is getting a little dry here. That's better. these assays don't always agree with each other. So particularly there are cases where the transduction inhibition assay or the cell-based NAB assay can give a fairly low signal where you're unable to detect antibody, and that's what's being shown here. This is some data from the sponsor Unicure, and what they had shown in their early clinical trial, they had a less sensitive GFP-based neutralizing antibody assay, and you can see with this less sensitive assay, they actually had better correlation. When they got into the positive range, they were able to detect antibody. When they switched to a more sensitive luciferase-based assay, you can see down here that there's some positivity in the assay where you're unable to detect antibody. So a discrepancy here in the results. What you would really like to see is this good correlation here where you're both NAB positive and TAB positive, but you don't always have that. And this is another example from Felice et al. From several years ago in hemophilia patients. This is basically 100 healthy donors plus 24 hemophilia A patients. And what's being shown here is there is some concordance with both the TI assay and the TAB assay, and these axes are reversed relative to the previous slide. And so these are patients that were excluded from the clinical trials. Patients that were included in this case were basically double negative. And as you can anticipate, in some cases, you can have these binding antibodies here, or total binding antibodies that don't have any neutralization capacity. What was a little more surprising is when you saw this neutralizing titer down here in the absence of any detectable antibody. You can see that as these titers get a little higher, then you are detecting antibodies. So at the low range of these assays, you seem to get into the situation where you're getting some neutralization on a plate in vitro, but it doesn't necessarily correlate to antibodies that are detected. And this kind of led to a question that what is the right answer here? If you're going to use these assays to try and pre-select patients for enrollment into your clinical trial, which assay should you use? Which one is going to give you the best information about which patients are likely to respond to gene therapy? And so one way we tried to answer that question was to do a non-human primate study that had evidence of both neutralizing capacity and also binding antibody. Just really quickly here on the left, there were 4 groups of animals. Group 1 was negative in both assays, the TAB and the TI assay. Groups 2 and 3 were very similar in that they had both negative in the TAB assay, so no detection of any antibody, but some amount of positivity in these neutralization assays. And then group 4 here was double positive, so good concordance, both detection of neutralization as well as binding antibodies. You can see it was really this group that had less efficacy, with the exception of this one outlier in particular. This is looking at the expression of factor VIII protein from an AAV5 gene therapy vector. Here is the protein AUC, and then the liver transduction here on the right. You can see with all these different measures, group 4 here, where you're able to detect antibody. The only group that you were able to detect antibody was the one that had inhibited efficacy in these groups of animals. And this led to BioMarin basically switching to the TAB assay as an enrollment criteria and then moving forward with that assay as a companion diagnostic that was contemporaneously that's a tough word, approved along with the factor VIII gene therapy. And so one question that comes up a lot was, why isn't there more standardization for these assays? And I think in answer to that question, there is a lot of standardization. With respect to the methodology of how you do these assays, there's been a lot of, wrong button again. There's been a lot of publications, mostly going back to Gobi Shankar's paper in 2008, on how to develop particularly ELISA assays to detect anti-drug antibodies against protein therapeutics, and then that's been carried forward in some of these other publications looking more specifically at assays that measure antibody to AAV. So there is a lot of information out there about how you go about establishing the cut point of these assays. How do you determine somebody who's positive versus negative? What's less clear is how you apply this to the clinic. And by that I mean, even though you've detected antibody, at what level of antibody do you no longer get efficacy in these patients, or at what level of antibody does this become a safety issue upon dose administration? That's less well known. I'm going to illustrate that with 2 different sort of diametrically opposed approaches to 2 hemophilia gene therapies. One is Roctavian from BioMarin for hemophilia A. In that gene therapy clinical trial, all antibody-positive individuals were excluded. That contrasts with Hemgenix for hemophilia B for that phase 3 clinical trial. It basically accepted all comers based on that data that I showed you previously, where they were able to use a more sensitive assay to determine that they had already dosed some patients with pre-existing immunity, and those patients were fine. So they moved forward with that in their phase 3 trial. So a little bit about how we establish these cut points for these assays. What does it mean to have a positive response? The way that's generally done is to take 50 to 100 healthy donors, run them through various iterations of your assay to try and eliminate the ones that are outliers and are likely positive. As we saw from the seroprevalence studies, 30 to 40% of people are generally going to, at least, are going to have antibodies to any particular capsid serotype that you're looking at. So you establish the cut point. That gives you this curve here, basically. That gives you the antibody negatives. Over here on the right are the antibody positives. And what's being shown here is basically that there's some overlap down here. And this is where things sort of come into question and make it a little bit less clear. So the clinical decision limit is something you can think of as an antibody titer threshold, below which you're able to dose and above which you're not. And this is where the ambiguity comes in, is how do you set that clinical decision limit actually in the clinic, particularly in the absence of any empirical data that tells you how to do that. And the answer is that that's really hard to do, and that's why I think we're a little bit stuck in some of these cases. What's being shown here are these action limits where you might set some sort of titer threshold above which or below which you do or don't want to dose. And that really needs to be determined using empirical data. And it's that empirical data that's been kind of lacking in the clinic to be able to make those kind of decisions. So comparing these two gene therapies, Roctavian on the left, the pivotal phase 3 excluded AAV tab positive. There was contemporaneous approval of a companion diagnostic. From the label, commercial drug eligibility criteria includes no antibodies to AAV5, and again, this was approved as a companion diagnostic. Conversely, the Hemgenix trial, also AAV5, also made in insect cells, so very similar gene therapies, just one encoding factor 8, one encoding factor 9, did not pursue a CDX or an LDT. The pharmacodynamic profile was not significantly different in patients with AAV5 NAbs up to a titer of 678.3, that's very specific. And pre-existing neutralizing AAV antibody titers, they say could impede expression at desired levels. So what did that actually look like in the BioMarin trial? What's being shown here are the 134 patients that were dosed in the phase 3. And two different things I want to show here. One is that there were 3 patients that screened negative in the companion diagnostic assay, but then were remeasured again at day 1. So during screening, they were negative, but at day 1, when they were remeasured just prior to dose administration, 3 of the patients did test positive in the assay at titers right at the MRD of the assay, 56 and 91. You can see down here the mean factor 8 activity in those patients was a bit less than what we saw in the mean for the overall population. So these 3 patients were positive at day 1, a little bit less factor 8 expression. Conversely, looking at results in the neutralization assay, so this wasn't used for screening, but was only measured at day 1 and then after dose administration. There were 11 patients of the 134 that screened negative in the TAB assay, but were positive in the TI assay at day 1. And those 11 patients actually did just fine. So you can see here the mean and median factor 8 activity relative to the overall intent to treat population was actually just a little bit higher. So this kind of recapitulates what was seen in the non-human primate study, where it was only animals that had evidence of binding antibody were shown to have limited efficacy in the non-human primate trial. That was recapitulated here in the phase 3 clinical trial as well, kind of reinforcing the idea that it's the antibodies that are positive, not necessarily the neutralizing activity as measured in a cell-based assay. And similarly with Hemgenix, also using a cell-based NAb assay, you can see here that their patients that were NAb negative had mean antibody titers of about 41 versus those with NAbs of about 32. I won't elaborate on this too much. It was shown before. As Kathy said, what's great is that these are both in the efficacious range, so very helpful for these patients. So I'll stop there and just summarize. Pre-existing antibodies, they vary in seroprevalence and titer across capsid groups. Both binding antibody and neutralizing antibody can impact the efficacy of gene therapy dose administration. Total antibody assays may be more appropriate for screening methods for exclusion and better predict in vivo efficacy. There are existing guidelines for standardization of antibody assays, but how you apply these to the clinic and how you determine a clinical decision limit or titer threshold still needs to be fleshed out right now. The only way to do that is to collect empirical data, but that's really difficult to do in human patient populations. So yeah, I'll end there and I guess we're taking questions at the end, so we'll pass it off to the next speaker. Thank you.
Okay, our next speaker is Tomas Baldwin from Great Ormond Street Hospital, where he obtained his PhD. Do I see Tomas in the audience?
I'm here.
Ah, yeah, good. Okay. And Guilford Street Laboratories, which he co-founded. And he's going to talk to us about applying LC mass spec to getting a more precise characterization of antibody titers.
Thank you. Okay. Okay, we on? Yep. Okay, thank you for the introduction. Can we all hear me? Yep, brilliant. And thank you for the invitation to talk today. So I'm Tom Baldwin. I'm a paediatric biochemist at Great Ormond Street Institute of Child Health in London. And I'll be talking a little bit today, as just mentioned, about mass spec and how that might be able to help solve some of the issues we have with assessing seropositivity with AAV. So no relevant disclosures here. So before we get into AAV, take you back a little bit to early 2020. Obviously SARS-CoV-2 sweeping across the globe, lots of governments locking down, including that of the NHS. And our team was seconded by the UK government to develop new, faster, better testing methodologies, not only for the virus itself, but also antibody response against the virus. And this is what we came up with. So very heavily leveraged the playbook from an ELISA. Quite simply binding spike protein, in this case, from COVID to wells of a plate, incubating with a very small amount of patient serum, so we're down to 15 microliters per sample. Now, instead then of adding your secondary antibodies and your calorimetric or fluorescence assays, we're proteolytically digesting and then analyzing on a mass spec. So we're going from sample to data within about 3 hours. Now, the benefit of doing this via mass spec is that in one assay, we're collecting fully quantitative data on all antibody isotypes and subclasses, but also the complement pathway as well, and deposition and activation of complement, which we've heard quite a nice amount today about, and I'll touch on a little bit later. We then validated this fully on a really substantial cohort of healthcare workers in the UK, noticing that we were able to accurately pick up differences in the immune complex after exposure, vaccine or infection or both, and really deeply characterize this response, which we can sort of see down the bottom here, and how that profile changes and what that potentially means in terms of functional consequences. As you're doing with, as you need to do with a new sort of platform technology, we compare against the gold standard. In this case was the Roche anti-spike assay, and we can see whichever sort of criteria we're looking at here, so total immune complex, IgG1, or a total IgG, we're correlating really nicely with the gold standard, but obviously we're collecting all of this information in one go instead of multiple assays. And importantly, as you can sort of see here, we're looking at multiple different variants of concern. The data we're collecting is correlating with functional outputs, so live virus neutralization in this case. So we felt that we had developed something really quite powerful here. High throughput, fully quantitative, correlates really nicely with the gold standard, and a very good correlation then with functional outputs. We're measuring things that matter. So then we're ready to roll this out to the UK and EU and integrate into healthcare. And it was precisely at this point the UK decided we no longer wanted to test for COVID, typically. So we left with what we felt was quite a powerful analytical tool and nothing to do with it. So after conversations, obviously within our own team, but also the wider UCL network on different opportunities to expand here, AAV seropositivity was one of the big questions that was raised, whether we could apply the platform here. Now, we've heard a litany of things today about seropositivity and issues, quite nicely documented by Brian then, about different methodologies and standardizing and implementing that. So I won't go into that. So the question for us was, can we take the platform we've developed and adapt this for AAV seropositivity, so a fully quantitative, high throughput assay? Now, fortunately for me, the platform is quite robust, so it was nearly as easily as just switching the bait out at in the interim. So instead of binding SARS-CoV-2. We're now just binding AAV to wells of a plate or beads. In this case, AAV9. And again, now we're going from sample to data within a period of about 3 hours. Now, the benefit of doing this by mass spec, as I mentioned, we are quantitating in absolute amounts here. So we're able to start reporting out nanograms of antibody per nanograms of bait. In this case, it's going to be AAV9, which importantly then allows us to start generating things like reference ranges for what is and isn't positive. That allows that to be quite easily standardizable between different laboratories. And another highlight of doing this is that we can also then quantify the bait binding per well, which can vary across the plate and lead to sort of issues with variability intra-samples. So we're able to correct for that also. So the platform was established, we're able to measure AAV, we're able to pick up samples. We then started a collaboration with Professor Francesco Montoni and Baronello in Great Ormond Street and their teams in order to assess how useful this would be in a patient population of spinal muscular atrophy patients. And this is pre and post Zolgensma, which is AAV9. So quite a small pilot study initially, so 15 patients, 9 controls, but longitudinal follow-ups on quite a number of the patients as well, with the aim of seeing can we measure functional differences in the immune complex of these patients pre-post treatment. We've also done some work on inflammatory status as well, which I'm not going to get into today. So first port of call for us is assessing this sort of idea of seropositivity in healthy controls. It's obviously looking at pediatric patients here. Now, just looking at IgG1, we can detect a significant difference between patients who haven't received therapy. We're about 58% seropositivity here, which is a little bit high for AAV9, but quite low numbers at the minute, so we're bolstering this at the minute. But what it allows us to do is obviously start generating preliminary data on these reference ranges, particularly for what is a negative, quite importantly. And after this, now apply that to the wider cohort of patients. So collecting data on IgG1, really interesting to see that, probably as you'd expect, completely reactive IgG1 response to therapy. So post-infusion, all patients responding with a positive IgG1 response. No clear relationship with things like IgG2. Found patients both seropositive and negative pre-post infusion. And IgG4 is a little bit less clear. We've actually found no patients to be sort of seronegative by the current cutoffs we're using. Now, IgG4 is a little bit promiscuous in terms of what it'll bind to, so this is potentially indicative of things like infection with other AAV capsids or else. But what is quite important to note here is that this positive binding of IgG4 would contribute to a total IgG ELISA. So it might not be functionally relevant, but contributing to what could then be determined seropositive in certain patient groups. What was quite an interesting finding in this patient cohort is when we're assessing these samples longitudinally, the IgG1 response is escalating over time. We're only going up to 7 months here because that's the duration we have, and obviously we talked earlier about this will go up to 14 years, which is incredible, but it only seems to be happening with IgG1. Now, we've checked all IgG2, 3, 4, doesn't happen. So an IgG1 specific phenomenon here, but it also correlates really perfectly with increased complement deposition onto the immune complex. Now, we've checked this from C1QC all the way through to C9, a perfect correlation, again, now only with IgG1 and not other complement activating antibodies. So increased IgG1 response over time, increased deposition of complement, and again, only with IgG1. Now, quite important, we didn't actually observe any non-antibody mediated complement deposition in this study, but again, quite small scale at this point. So the million-dollar question for us is, we're accurately able to now quantify things like antibody binding, complement deposition, but the big question is, can we predict which patients would now go on to have a severe reaction to therapy, hospitalization, hepatitis, these sorts of things, and eventually things like therapeutic efficacy. The pilot study we've done so far is not particularly suitable for this because of the numbers. Only 2 good patients having pre and post that we've used here. But if we were to look at these 2 patients, patient 1, an expected reaction, so mild inflammation, slightly raised liver enzymes, and if we look at the binding profile, just a little bit of IgG4 doesn't seem to have an impact on the patient. and then post-infusion having that reactive IgG1. Patient 2, however, had a very severe reaction, so hospitalization, hepatitis, significant inflammation. And their pre-infusion panel here, we are actually detecting binding of multiple different antibody isotypes and associated complement. Now, this patient obviously passed the criteria for entry to the clinical trial, so difference is obviously what we're looking at here and what might be reported previously, but again, reactive post-therapy as well, so that IgG1 signal increasing. Now, obviously, this is really small scale at the minute, but starting to notice differences potentially between patients who've responded well and not, but a lot more work to be done here. And finally, the question we were asked by the team at GOSH was whether the platform would be suitable to start looking at things like cross-reactivity, which we've talked about today already, with the aim of looking at IgG1 response. In this instance, we've done AAV 8 and 9, because it's what we had to hand. But really interestingly, finding that cross-reactive response between AAV 8 and 9 for IgG1. IgG1 not really, not commonly cross-reactive, at least in protein therapeutics, but what is really cool is that the IgG1 response against AAV9 is complement activating, but against AAV8 it's not. And whether that's to do with quantity of binding or something else, this is what we're investigating at the minute. But again, only really starting to get into this investigation now, but the ability to assess cross-reactivity seems to be working quite nicely as well. Now, I'm conscious I've whizzed through that for timing, but as a conclusion, we've adapted our LCMS platform for SARS-CoV-2 to AAV9 in this case to look at changes in immunocomplex in SMA patients pre and post-treatment. The anti-AAV9 response in all patients was found to be responsive for IgG1. It's a little bit less clear with other subclasses and isotypes. And this IgG1 escalating over time with a really strong positive correlation with things like complement, which we don't observe elsewhere. So it's starting to look at putting plans in place to generate data on how this expands in much larger cohorts as well. And that's the next steps for us now. So much more data, generate fully quantitative reference ranges with the idea of being able to roll that out in an easily standardizable manner. So thank you for your time today. Big thank you to the teams at UCL and Great Ormond Street, particularly Dr. Wendy Hayward, who's here with me today, and Professor Kevin Mills. And I will look forward to taking questions later. Thank you very much.
Okay, thank you. That was super interesting, and I think you're going to get a lot of questions. So our next speaker is Dr. Sharon McGonigle of Solid Biosciences. She's going to be talking about anti-AAV seroprevalence in a DMD cohort and assessment of impact on efficacy preclinically.
All right. Good morning, everybody. Can you hear me okay? No? Oh, can you hear me now? Hello. Yeah. Good morning, everybody. Can you hear me now? Yeah. Leave it up? Okay, perfect. Awesome, thank you. So good. You're going, okay. All right. Good morning, everybody. Thank you so much for the invitation to be here at this amazing day of science, Michelle. Really appreciate it. So I work at Solid Biosciences. I'm going to present a small vignette today of the work that is generated by our immunology team there. Kriti Patel, who's in the audience today, leads that team, working with a small group in the R&D group, generated this data. And so I'm really excited to present today on their behalf. So you've seen our forward-looking statements before. The overview of my talk, I'm going to introduce DMD and SGT-003, and then I'm going to talk about the seroprevalence in our DMD cohort, really talk about the determination of seropositivity, thinking about that titer cutoff and the tools that we have to look at efficacy thresholds using preclinical studies, and then I'll just list some of the mitigation strategies that we're thinking about for AAV seropositivity. So Annie earlier gave us a beautiful and really eloquent discussion of how devastating this disease is for the patients and the families. And I'm not going to go through all of this background here today, but this is obviously why Solid Biosciences came into being. This is an incredible focus that we have at Solid Biosciences and that we continue to have to try to develop drugs that will really impact this disease. So again, here's all the background. The only thing I will highlight is the unmet medical need. And despite the approval of Elevidys, which is a gene therapy that treats Duchenne, we still believe that there is a significant unmet medical need, and particularly for individuals with DMD who are seropositive, and that's obviously the group that we are here to discuss today. Okay, so now I'd like to introduce you to SGT-003. This is our next generation AAV microdystrophin for treating Duchenne. It is composed of our novel muscle-tropic capsid, SLB101. SLB101 is based on an AAV9 backbone. It contains a 7 amino acid peptide insertion that has an RGD peptide. The data here shows the muscle tropism. We're looking at biodistribution in the quadriceps of mice, and you can see the increased biodistribution of SLB101 over AAV9. And we also see some liver detargeting with less SLB101 in the liver versus AAV9. And so those biodistribution, muscle tropic effects also translate to increased muscle expression of the transgene. And the transgene in SGT-003 is a unique microdystrophin amongst the clinical microdystrophins in that it contains this R16, R17 nNOS binding domain. This domain then is responsible for recruiting and restoring nNOS, or nitric oxide, to the muscle membrane. That can help prevent exercise-induced fatigue and also prevent or help with muscle damage and muscle injury. Okay, so SGT-003 is now in our first in-human study, and this study is called Inspire Duchenne. This clinical study started earlier this year. As per our press release, we have now safely dosed our first 3 patients in this study. The clinical dose is 1E14 vg per kg, and we're really excited and looking forward to the first quarter of next year when we'll be releasing the data on those first 3 patients, and that will be safety data, biomarker data, as well as expression data from that 90-day biopsy. Okay, so now I'm going to switch gears and talk about seropositivity. I'm going to really quickly skip through all of this because you've heard a lot of it this morning in some really beautiful talks. We'll talk about the fact that we can actually detect pre-existing antibodies in newborns from transfer from the mother. These wane, but very quickly in early toddlerhood and childhood, you can see from that exposure to wild-type AAV that we're able to detect pre-existing antibodies. I'll move to the next one and sort of say, how do we define AAV seroprevalence? It's the frequency of individuals in a population who show antibodies to AAV and has already been described. So because we're looking at wild-type exposure, that seroprevalence can really vary by geography, by race, and by age. I'm showing you some really nice data that was generated by the Pfizer group, looking at the cross-reactivity across a wide range of serotypes. This is with a NAb assay, and you can see seropositivity here from 29 to 48% across this pretty diverse group of AAV serotypes. The other thing that has been really highlighted this morning is that the rate and the calling of a seropositive serum sample really depends on the assay, the details of that assay, as well as the titer cutoff, et cetera. We have heard a lot about the impact, so I'll quickly go through this as well. But the presence of those anti-AAV antibodies in the serum will just reduce the clinical benefit that a patient will get from a gene therapy treatment. They do that by binding and neutralizing to the AAV, and obviously the consequence of that is those patients are not able to be enrolled into studies and are not able to receive that clinical benefit. Others have spoken to the aspects on the slide, so I'm just going to keep moving and sort of start telling you about the data that we have generated in our cohort. So we were able to access serum from a cohort of 58 individuals with DMD. These donors ranged in age from 2 years old up to 18 years old, with an average age of 10 years. The serum samples were obtained from a biobank that is curated and maintained by Cure Duchenne Link, and we're extremely grateful to them for generating this biobank and for us being able to access it. I don't have to go into these assays at all, thanks to the beautiful talk that Ryan just gave us earlier, and he has the most amazing graphics that I've ever seen. So that really negates the need for me to go into this. So I will just say that our total antibody assay, here we're using a capture-based ELISA method, as he described, and our secondary antibody is directed against IgG. So throughout the rest of the talk, I'm going to be referring to TAB IgGs measured by this ELISA. And again, our neutralizing antibody assay or transduction inhibition assay is very similar to that one that Brian so beautifully described. So the first thing we wanted to show was how these assays are, what data do we get in these 58 samples with these two assays. And so what we're showing here is TAB and NAB titers from AAV SLB1 and AAV9. And here on the y-axis are the NAB titers, and on the x-axis are the TAB IgG titers. And you can see this really nice correlation for SLB101 and similarly for AAV9. And so given this nice correlation that we're seeing with our assays in this particular cohort, we are now, and for the rest of the talk, I'm really just going to only talk about the TAB IgG data. So one of the first things we wanted to look at was the cross-reactivity, and the cross-reactivity between SLB101 and AAV9. We also included R874 because this is the serotype that is in Elevidys. Again, that's the gene therapy that is approved for Duchenne. So first of all, we saw in our TAB IgG 100% cross-reactivity with AAV9 and SLB101. Maybe not unexpected, given as I've said that SLB101 is based on AAV9, but this was important data for us to generate, and it was nice for us to see this result. What was super interesting was the really high levels of cross-reactivity with R874, 98%. And then when we looked at the titers across these three serotypes, we could see that the titers varied only by 1 to 2 dilutions across the three AAV serotypes. If you squint, you can see that some of these titers actually look really high, which again was a surprise to us. And so I've plotted a different way here. So what you're looking at in this graph is we've taken those 58 patients, we've evaluated which ones in that 58 are seropositive, and now these are plotted based on the titers that we observed. And so what was really interesting is about 50% across all three serotypes have titers that are greater than 1 in 6,400, which is the highest titer that we tested. Certainly for me, this was pretty surprising. Again, we're dealing with a pretty young population here, and I was expecting most of the patients to be in that low to mid group. So this was a bit of a surprise. When we looked at the age group here, we were able to see, there's quite a spread. But of these donors with the titers, these high titers at 1 in 6,400, the mean age was 8 to 10 years. Okay. And so one of the big questions, and Brian really nicely spoke about this as well, one of the big questions that we still have is titer cutoff selection and how we use that to determine patient eligibility and patient enrollment in our studies. And I think it remains a big question in the field. Every sponsor does it differently, as we know. We all use different assays, different technical components in those assays, and so it makes it a really difficult question. But I thought I would walk you through an example here of how the titer cutoff and the selection of that can really impact the patient enrollment. So this is the data that I've shown you earlier. This is data that's showing seropositive, the amount of seropositivity at the highest stringency that we can look at. So this gets back to that statistical cut point, and basically anybody who is seronegative here, we cannot detect antibodies in their serum, and so our expectation is that they will gain clinical benefit from our gene therapy. So let's say, for example, because I had to take all of the numbers off here, but let's say that this is a 1 in 50 titer cutoff. If we are then able to lower the stringency of that titer cutoff, say to 1 in 200 or 1 in 400, you can see now that you lower that seroprevalent population. And so now we have this group of patients here that are denoted by this arrow that are now potentially eligible for enrollment into the study. And so the big question that remains then is, will this patient population, which have low titers of seropositivity, will they gain clinical benefit from our gene therapy? And so one way that we try to look at that is by doing these preclinical studies to evaluate titer cutoff thresholds for efficacy. So I'm showing you one of the studies here. This is a mouse study. So this mouse is the mice are they're passively transferred with human serum, so that we end up with mice with levels of seropositive titers from low titers all the way up to high titers. We then administered SLB101 that's expressing a luciferase reporter, and at the end of the study then we can measure luciferase activity. And so here you can see this really nice dose-responsive loss of luciferase activity in response to the increasing titers. So at a high seropositive titer, you see that we have absolutely lost all luciferase activity, whereas at low titers, you can see that the luciferase activity is really similar to a seronegative mouse. So this again is really helpful data. What's really interesting about this is what I'm showing you is whole body IVUS. When we actually take out and look at different tissues, we can see quite different results. So heart tissue, for example, seems to be more resistant to higher levels of seropositivity, whereas tissues such as the diaphragm or skeletal muscle is more susceptible to lower levels. Again, so getting to the point that everybody has made so far that the dose is important, the target tissue is important, as we're doing all of these studies. So then I'm showing a graph that is very similar to the one that Shari showed right at the beginning of this morning, where we're trying to really stratify our patient populations based on these titer ranges. So the titer that we say, based on our assays and our MRD is eligible is here. And then based on our preclinical threshold studies, we may have the potential to identify additional low titer patients that could benefit from our AAV therapy. When we then start to talk about this mid to high seropositive patient population, we're going to need mitigation strategies. And potentially we're going to need combinations of those mitigation strategies. So we at Solid, and probably everybody else in the room, are thinking about the various different ways that we can mitigate IgG through IgG depletion, plasmapheresis, FcRn inhibitors that have already been mentioned, all the various IgG degrading enzymes that are out there now. And then future efforts would obviously involve trying to genetically engineer their capsids so that they can evade the immune system, and for redosing, obviously, immunomodulation to try to attenuate antibody production. These are the conclusions. I think in the interest of time, I don't need to go through all of these. But basically, our goal at Solid is really to try to find the technologies to mitigate pre-existing antibodies so that we can expand the addressable patient population. And with that, I would just like to acknowledge again the team at Solid that generated all of this data. Again, Kuranda link for the access to this really valuable resource. The individuals with DMD who contributed to the biobank, and our DMD patients, their families, caregivers, our clinical investigators, and everybody associated with our Inspire Duchenne clinical trial. And thank you for your attention.
Thank you for staying on time. Thank you. Okay, our next to last speaker in this session is Dr. Robert Calcedo. He has long history working in the field, primarily in preclinical work and immunology, and specifically in antibodies to AAV. So he's going to be talking about durability, cross-reactivity, and window for dosing. Dr. Calcedo for many years was at the gene therapy program at the University of Pennsylvania and now is in charge of preclinical and immunology at Affinia Therapeutics.
Part of my presentation now. Okay, here we go. So thanks, Kathy, for the kind introduction. And you're going to see that a lot of my slide overlap with what others have said. And I'll try to focus just on those differences. By now everyone knows that seroprevalence in the human population, AAV, is very clear. It doesn't matter where region in the world you go, you're going to find antibodies, especially neutralizing antibodies to every single serotype. And historically, AAV2 was the first serotype used in gene therapy, and it's also the serotype that are usually higher in seroprevalence and magnitude. Now, a question that arises at the very beginning is, what we see in healthy subject is also what we see in our patient subject, and more or less, it could be some differences based on the disease, but that prevalence that we see in healthy is still sustained, and especially on the right at Duchenne muscular dystrophy subject, AAV2 remains the serotype with a higher prevalence compared to other AAV serotypes, and most of them are already in the clinic. Now, what is interesting, especially for those that are working on engineering new capsid, is that actually we have AAV capsid that are not recognized by the immune system, and one of those is rhesus 3233 that belong to the AAV4 family. And across, almost 1,000 samples that we tested back then, almost only 2-3% of the population was seropositive for that capsid. Now, what is interesting about this capsid, and Sari mentioned before, and it's very immunogenic, she used it in her studies, is that, by modification of the capsid, and this is a naturally occurring capsid, you are biasing, changing the tolerogenic properties of AAV. We all know that, transgene responses are very low, usually very low when we use AAV. But in this capsid, for example, responses to the transgene are very high. So this particular serotype becomes, or is kind of like an adenovirus, very potent vector for inducing immune response and for actually vaccine development. So there are naturally occurring AAVs that have a low seroprevalence in the population, but we don't use them because they are very immunogenic. So something that also have been mentioned before is, well, these responses happen in the adult population. They are there, but when they actually develop. And early on, when I was at University of Pennsylvania, we conducted this study, basically characterizing the pediatric population, and we can clearly see that, very early on, antibodies are transferred from the moms to their babies, and that response over time, goes down, and that's a good opportunity for gene therapy. Similarly, in Duchenne muscular dystrophy patient, we have this different kind of plotting the data, but you can see early on you have less prevalent for both AAV8, so patient with fewer neutralizing antibodies. So that said, it brought an important question to us. We see all these responses in humans. Some are positive, some are negative, and when that immune response develops, and so we conducted this study, and this is a retrospective study in chimpanzees where we have samples that span 10 years. And actually what we found is that, very interesting, some animals, and chimpanzees are animals that are not in cages. So they are in an open field, so they are social animals, so kind of like, like humans. So, it kind of mimic the natural immunity you may develop in humans. And actually we found chimpanzees that were, for a period of 10 years, they were negative for neutralizing antibodies. Some others that were chronically positive for neutralizing antibodies. And some others that we capture, when they become, we call them acute, when they become positive for neutralizing antibodies. What was also interesting is, that the study was conducted not only with AAV8, but also with other serotypes, and we looked for cross-reactivity. And so those animals that were negative for AAV8, they were also negative for all the other AAVs that we tested, and belong to different clades of AAV. Now, those that were chronic or acute, they developed broadly cross-reactive antibody responses, and these are natural exposure to AAV. So under natural conditions, chimpanzees and by comparison humans, they develop very cross-reactive responses to most of the AAV serotype that we tested. So that was very interesting. So we went back to humans, and now this is not a novelty. We know that antibody responses, natural immunity to AAV is very cross-reactive. Even when you're using viruses, AAVs from different clades, a clade E, AAV2 is down here, clade B. So there's some cross-reactivity. The magnitude is lower, for sure, but the cross-reactivity is there. So if the magnitude is lower, I mean, using different AAV serotype, we can overcome the primary response or a response to, for example, in this case, to AAV2. So that's what, determine, that titer, that the magic number is so important. What is the cutoff? Where do we determine if 1 to 10 or 1 to 20 neutralizing antibody is important or not? So another interesting data, this is coming from a clinical trial conducted for alpha-1 antitrypsin using AAV1, and relate also to cross-reactivity. So in the previous slide, it was natural immunity. Now this is immunity or immune response after transgene administration. And what is interesting in this study is that they injected a subject with no neutralizing antibodies, and we have 2 doses. And the response on those patients that were negative for neutralizing antibodies, very specific to the vector injected. Long term, even 2 years later, you still detect neutralizing antibodies. Now, what was interesting is that a couple of subjects have positive responses against AAV, but they were administered because it was intramuscular injection of the vector. Now, they developed really highly cross-reactive antibody responses to every single serotype. So what is interesting is that, by injecting AAV1, you're able to boost memory responses that are broadly cross-reactive. Antibody responses are not monoclonal, they are polyclonal. You're not only raising one antibody, but a population of antibodies that have neutralizing properties. So that's also an interesting finding. And I think one of the important studies that determine as how important neutralizing antibodies are in gene therapy, especially for intravenous administration, was in this preclinical study with cynomolgus and rhesus macaque using AAV8 and a dose of 3E12, basically targeting the liver. And as you can see, so these are naturally occurring neutralizing antibodies. So we have around 20 monkeys that we're pre-screened and categorized based on neutralizing antibody responses. And you can see that when you don't have neutralizing antibodies, less than 1 to 5, limit of detection, liver, is very good transduction. And then when you start having neutralizing antibody, you see a reduction on the liver transfer and actually an increase on the number of genome copies in the spleen. So that is, and actually correlate with the lack of transduction in the liver. So, clearly, low level of neutralizing antibodies, can jeopardize your gene transfer in the liver, but also change the biodistribution of the vector, ending up in the spleen. So now this data, it all has to come together because the sensitivity of your neutralizing antibody, also is going to correlate with the dose that you're administering. This is a very low dose that we are injecting. So we can say that 1 to 5, 1 to 10, based on our neutralizing antibody assay that use an MOI of 10 to the 4, and injecting 3 times 10 to the 12 VGs per kg, you get neutralization. So I think that you can overcome that if you go higher in a dose. So that's why defining that titer depends also on your NAB assay, the dose you're planning to inject, and the quality, purity of your preps. And something that people mention a little bit in patients is seroconversion. Seroconversion is an important topic issue in preclinical in the clinic. You usually screen your monkeys or your patient a month before going into the clinic. And during that period of time, seroconversion can happen. And that's why collecting sample of day 1 is really important. But that is something that it's always complicated, and it's especially complicated when you're working with non-human primate. Now the topic of neutralizing versus total antibodies, I think I'm not going to repeat what Brian said. Really good characterization. What I want to emphasize is that, and this is just a limited number of samples, we have a lot of samples that I highlighted here. The graph on the right is the same one on the left, but just emphasizing those low level of neutralizing antibodies. So basically, we don't have neutralizing antibodies here. We have 6 samples with no neutralizing antibodies, but we have total binding antibodies. And if you're setting up your exclusion criteria on total antibodies, well, you determine whatever your criteria is, but it's 500. You're excluding 2 patients from that. That is, significant number of patients you're excluding from your clinical trial. So I think, the neutralizing antibody assay is definitely the one at least that should be used for inclusion criteria. You can have both, neutralizing and total antibodies, but definitely neutralizing antibodies is an important assay to characterize the population that is getting into the clinical trial. And then I'm not going to talk much about this, but yeah, route of administration matters. You can inject, in this case with monkeys, with high level of neutralizing antibodies, 160, and to the orange one, and you still have comparable level of transgene expression, with animals that are less than 1 to 5. When you go higher than that, you compromise, your efficacy. That in IV administration is as low as 1 to 10, 1 to 5. It can compromise your efficacy. And local administration in the CNS. So there was a few clinical trials, and I think Dr. Bonneman will talk about later on about the GAN clinical trials. So I'm not going to spoil, his talk. But, in that clinical trial, the route of administration was local, intrathecal, and patients that were positive for neutralizing antibodies were included. Why? Because you don't have neutralizing antibodies in the CSF, even when you have them in the serum. So these are the patients at different doses, and all of them were negative for neutralizing antibodies in the CSF. The dotted line are those that were positive in serum. Of course, you are going to, a lot of the vectors escape the CNS through this route of administration. So you're going to have not only a local immune response to the capsid, but also systemic immune response in the serum that is very high, comparable to IM or IV, even higher than intravenous administration. And these responses last for at least, for up to 5 years. So we will talk about, if there's a reduction on NAVs, well, they don't happen easily. A 10,000 NAV titer is a very high titer after 5 years. So just summarizing, what I said, the route of administration is critical to determine the importance of your neutralizing antibody data or your total antibody data. And neutralizing antibodies are not only total antibodies, could be N, total antibodies, should be used for patient exclusion criteria. And naive subjects develop less reactive neutralizing antibodies responses. And if we're thinking about, readministration of the vector, that could be really important, coming up with a different vector, different capsid. And prevalences of neutralizing antibodies, serotype and age dependent, and neutralizing antibodies are everywhere, long-lasting, and difficult to modulate, even in the presence of a strong immunosuppression regimen. And because I didn't mention before, but in the GAN clinical trial, they immunosuppressed. There was a triple immunosuppression, and still you develop strong antibodies to the capsid. Thank you so much for your attention, and we'll take your question after.
Okay, so our last talk of this session is by Juliette Hordeaux. Juliette is a board-certified veterinarian and also has a PhD in translational sciences. I think she studied with Philippe Molière, right? Okay, and more recently has been at the gene therapy program at Penn for a number of years now, and she directed many of the preclinical studies, particularly in large animal models and non-human primates, and is going to talk about the impact of AAV seropositivity in some of those studies.
Thank you very much, Kathy, for the introduction, and thank you for the kind invitation, Michelle. So as the last speaker of the morning session, I'm going to be hammering down a lot of notions and concepts that previous speakers already made beautifully. But as a translational scientist, I know that both non-human and human primates learn better through repetition. So I hope you won't mind the redundancy. All right, this is our disclosure. So all the data I'm going to present were generated at the gene therapy program with various funding sources, and I am now an employee of Gemma Therapeutics. So these are the concepts and notions I'm going to cover very rapidly. General differences between non-human primates and humans' immune systems, the impact of AAV seropositivity on both efficacy and safety, and very quickly examples of immunomodulation strategies. So first of all, the differences between human and non-human primates. The polymorphic major histocompatibility, okay, I'm going to say MHC. The polymorphic MHC locus encodes cell membrane-bound glycoproteins that regulate the immune response through presentations of antigen. And it's divided into classes. Class 1 molecules present intracellular antigens, and they are present on the class 1 molecules are expressed on all nucleated cells. Class 2 molecules are expressed on the surface of professional antigen-presenting cells, such as macrophages, also endothelial cells, and they typically present extracellular antigens, and that's the relevant arm for generating antibodies through the secretion of cytokines that are going to stimulate B cells. As you know, macaques belong to the old world monkeys and are very close to humans phylogenetically. However, when you look at matched comparison and alignment of the MHC class 1 and class 2 genes, you can observe that in general, these are highly conserved. So you find the autologs genes between macaques, so between humans on the top and macaques on the bottom. But if you focus on class 1 here, you can see the orange blocks represent the polymorphic alleles for class A, B, and C for HLA, and they have equivalent genes in macaque. In this type, they're called MAFA genes. But you can observe that those autolog genes went through a lot of evolution with duplication, deletion, rearrangements, which lead to much more genes being expressed in macaques compared to humans. So a traditional haplotype in macaque will actually express 3 class A alleles compared to 1 in human, no class C, and multiple class B genes, sometimes up to 19 class B genes can be expressed in macaque, which leads to a higher diversity of haplotypes. The class 2 are much less diverse and much better conserved. So you can see those blue blocks between DR, DQ, and DP alleles. They all have orthodox genes in macaque, and only the DR genes are a little bit more divergent due to duplications. But overall, the class 2 are closely preserved. So let's focus on toxicology study. Because of the macaques are a close related species, they are heavily used in toxicology studies in biologics and AAV gene therapy, specifically for this close immune MHC relationship, even though, as I mentioned, it's not perfect. So as a general principle, what we observe in toxicology studies of AAV gene therapy is that the class 1 restricted T-cell and cytotoxic T-cell responses are typically more pronounced against the transgene product itself rather than the capsid. And this is probably easily explained by the fact that most gene therapy drugs for gene replacement strategies encode a human protein in a macaque. And even though, again, there's typically more than 95% amino acid identity in the proteins, you do have non-self epitopes that can be recognized as non-self and trigger CD8 cytotoxic responses. But responses to the capsid, CD8 responses to the capsid in macaque are not very prevalent and are less robust than in humans. So that's, right off the bat, that's an important difference between NHP and human responses. The class 2 restriction and the antibody responses, on the other hand, are much more similar between humans and macaque, and it makes macaque a very good model to study them. Here you can see an example of an AAV9 intravenous administration in macaque at a high dose, 2E14 genome copies per kilogram, encoding a therapeutic transgene or GFP. In that case, it's actually GFP. The IgG response kicks in really rapidly during the first week, and then the IgG response comes a little bit later during the second week. What about complement activation? There was a lot of comments and discussions around that from the previous speaker. Some complement activation and TMA responses have been seen in high-dose AAV9 clinical trials. So obviously, we were interested to study that and see if we were observing the same things in macaque. And even though we have this immune response with both IgM and IgG appearing in macaque, the complement activation response seems not the same as in human, and specifically the marker for classical activation. So the immune complex-related activation, C4a, is not activated in non-human primates in these macaques. And there is some activation at day 3 of the alternative pathway here shown as BB. And one difference between macaque and humans is that even though the complement genes are, again, highly conserved, monkeys tend to have more alternative pathway responses than classical, and humans tend to have more classical responses than alternative. So that might be one reason of the discrepancy here. AAV seropositivity prevalence, I'm going to very briefly present some comparison between NHP and human data. This is data generated by the GTP Immunology Core, Jessica Chichester, so I thank her for this slide. And you can now find Jessica and the Immunology Core under Franklin Biolabs. So in both humans and primates, we've seen nice data showing that the seroprevalent titers depend on population, depends on age, and it's pretty much the same in macaques. Depending on the age of the animals, you're going to have higher seroprevalence in older animals, and depending on the populations, specifically depending on if they are F0 wild caught or if they are purposely bred, you're going to have differences in seroprevalence as well. But in general, if you look here at a comparison for AAV8, AAV9, and RH74, you see that AAV8 has a high prevalence in both species, and it's pretty much balanced and the same seroprevalence with around 50 to 60% in both species, and relatively the same in rhesus versus cynomolgus macaque. AAV9 has an actually higher seroprevalence in rhesus macaque and cynomolgus macaque than in humans, so it can be tricky to procure non-human primates that are NAB negative if you're working with AAV9, especially if you're working with rhesus. And finally, the opposite is true with rhesus 74, where the very high seroprevalence can be found in rhesus and lower in humans. Sorry, I was making the opposite point for AAV9. The prevalence is lower in humans. All right, so what about the impact of, we talked about the impact of seropositivity on safety and biodistribution, and many speakers made the point that it is more an efficacy than a safety concern. And I'm going to fully concur with these statements and show you some data that supports that in non-human primates. This is briefly a study in which we gave high-dose AAV68 vector encoding a therapeutic transgene under a CAG promoter, ubiquitous promoter, dose of 5e13 genome copies per kilogram. Q68 is a clade F vector, highly similar to AAV9. So in our study design, we were enrolling on purpose some NAB negative animals and some NAB positive animals, and we had one low titer and one high titer. And we went into this study with the hypothesis that we were trying to replicate the TMA model and the toxicity that is seen in some clinical trials, with the hypothesis that if the monkeys had antibodies baseline, we might be able to trigger a more TMA-like complement activation toxicity. The results were not what we expected. First of all, looking at liver enzyme levels for the acute toxicity model, so we observed acute liver toxicity with ALT elevation that peaked on day 3. This is our cohort of NAB negative animals. And in our 2 NAB positive animals, we have our low NAB titer and our high NAB titer. They developed either no ALT elevation at all for the highest titer or moderate elevation for the low titer. And similarly, complement activation. So again, C4a, we were not able to elicit, again, any significant C4a elevation. This is the upper limit of normal. So no significant C4a, classical pathway activation, and the usual slide alternative activation in some animals. So this means that overall it was safe. We did not trigger any safety event in monkeys that had neutralizing antibodies baseline. And the reason for that, specifically for the liver injury and liver toxicity, can be explained by another paper I'm not going to have time to show you. But we observed that this day 3 liver enzyme elevation is highly correlated with expression. So when you give high dose vector, the expression is turned on really rapidly. And we observed a high level of transcript as soon as day 3 in hepatocytes and endothelial cells, leading to this early injury. So by preventing transduction, you actually prevent toxicity and safety. Well, you're going to tell me, what about efficacy then? If you prevent transduction, what's the point? Well, the point is that there is actually a sweet spot that can be found between the amount of neutralizing antibody, so between the titer, and the dose of AAV. So you can increase the dose, you can have some efficacy at some level. So again, this comes from the same study, same animal. We looked at cardiac transduction. This is an immunohistochemistry for the GAA transgene. Obviously, NAB negative animals have very robust cardiac transduction at this dose. AAV-Hu68 is cardiotropic. And if we look at our 2 NAB positive animals, the 1 to 20 titer did not lead to any decrease of efficacy in cardiac transduction, but it did at 1 to 160. So again, which threshold with the low efficacy depends on the route of administration and the titer of the NAB and on the dose of the AAV. And the 1 to 20 in that case would be both safe and would lead to some efficacy if heart is your target. Sorry, it seems to be frozen. All right, let's try. I'm trying to not push the button too many times because then it's going to zoom at the end. Can the team help me and unfreeze my presentation? Or maybe the battery. Oh, it looks like the battery is popping out. There you go. All right. So again, the route of administration, the point was made earlier. If you do an IM, if you do subretinal administration, there's no impact on efficacy. Same for CSF administration, as Roberto mentioned before, because of this different distribution of NAB between the serum and the CSF compartment. So when we give AAV through the ICM route, we observe good CNS transduction in this example. Yeah, I think the battery is not functioning anymore. All right. So in the CSF, this is a secreted transgene. We measure the levels in CSF post-administration. And in this study, the animals were either NAB negative or they were NAB positive through a passive immunization model using IVIG. And the efficacy was seen in all cases. And interestingly, in this case, it can be a strategy to prevent liver toxicity, because by injecting ICM and giving an IVIG dose IV at the same time, what you achieve is the same efficacy in brain, but you achieve total detargeting of liver thanks to the IVIG. So it's an interesting strategy to mitigate liver toxicity. So I'm going to need someone to advance the slide. I think this is dead. Oh, I can use maybe the... All right, I'll try to use that. It's not responding. All right. Brain administration, the bottom was about brain administration. So same, you see a multiple level of antibody titers with full efficacy after direct brain administration up to 1 to 160 titers, so pretty high titer. All right, next slide. A few examples, I will go very quickly of immunomodulation strategies to lowering the antibodies, the natural antibodies. So a lot of papers have been published. I'm showcasing 3 of them, and I'm not going to present everybody, Federico's data or others' data. I'm going to focus on one. So you can use neonatal Fc receptor inhibitor, and there's an approved drug to do that in order to transiently lower the neutralizing antibody. IgG cleaving peptidase is another strategy with similar transient decrease of the antibodies, and plasmapheresis is also another strategy. So my last data slide showing you the concept around these lowering NAB strategies is that an important notion is that none of them are lasting for very long. So you have to adapt the administration system and finding, again, the sweet spot and the therapeutic window where your titer is low, but it's going to bounce back very quickly. So if you need your AAV to be in circulation for several days in order to be efficacious, that might not entirely be working. But in this example, monkeys receive multiple rounds of this antibody against Fc receptor. And you observe that on day 0, which is the day where we want to give AAV, the animals in grid that received the treatment, compared to the animals that did not, you see a decrease in their total resist IgG level. And on the right, you can see that the anti-AVQ68 level are close to 0 by the time you want to give them vectors. So that can help. If you want, click 1 for the animation. I apologize for the animation, but that's showing you that the animal that had a 1-to-40 titer was able to rescue efficacy in targeting heart with the anti-FcRn antibody. I'm sorry, I cannot advance my slide. Here you go. All right, one more. That's my final slide, again, to hammer down all the concepts that I presented and that others presented this morning. Next slide, please. All right, next slide again. I should stop doing animations. So non-human primates and humans, MHC class and immune system are highly conserved. Monkeys also have similar levels of prevalence in seropositivity for some capsid, and it depends on age and population. NHP do not develop capsid immune complex-induced complement activation, so that's a difference with human. And importantly, there is no impact on pre-existing NAB on safety in any of our non-human primate models. The impact on efficacy depends on the dose, the route, and the titer. And really, I think by standardizing the assay between your preclinical and your clinical study, if you use the same neutralizing antibody assay, you can actually conduct some non-clinical studies in non-human primate that will inform the threshold at which you're going to enroll or exclude patients. And it's going to be data-driven. Several strategies are run to deplete NAB, and they're all transient, but probably by combining several, we could expand this transient period where the NAB titer are low enough to allow administering vector, which would allow to enroll and not exclude these patients with natural immunization. Thank you very much. Okay.
Okay, so we can go to 12:45. And can we have all the speakers from the last session up in the front? And then there are microphones up at the front for members of the audience who want to ask questions. And maybe I'll take the moderator's prerogative and get us started, because I don't see people at the microphones yet. So I'm going to start by asking Tom Baldwin, since you work at a children's hospital, do you have any information about clinical correlates of gradually increasing levels of complement in the children who got Zolgensma? So that was over about 8 months, right, that you detected gradually higher levels of complement proteins. And do you have a series of normal children who were analyzed in the same way? You guys must have done that.
Yeah, so, okay. It's something we're looking at now. I think the first objective obviously was to test that this was all feasible before we went into the wider cohort. But so it's ongoing, but it's absolutely something we're interested in.
Oh, okay, great. All right, well, if I don't see, is there? Yes, go ahead, Sharon.
So Tom, you showed IgG 1, 2, and 4, but missed IgG 3, unless I missed it somewhere, which obviously is very potent at doing multiple immune functions. Can you comment as to IgG 3?
Yeah, so this was just a bit of an attempt to not drown everyone in data, because obviously we're getting all of the antibody isotopes. So the IgG3, we didn't see anything striking there in terms of correlative with complement, or a very low amount of binding actually. compared to what we're seeing with IgG1 and 2 and 4. So just sort of focus there. But we're coming to publish this now, so all of the actual individual subclass and isotype data being included there.
And can I ask a follow-up question on just the assay format? So the complement that you're measuring, is that complement free in serum, bound to the antibody? So how is the complement being measured versus the antibodies?
So it's all measured in the same immune complex. So as you would with an ELISA, binding your antibodies, and then complement deposition onto that sort of immune complex then is what we're analysing, not necessarily digested from free serum.
Thanks. Hi. I guess a question for several of the panelists, and maybe Brian, you can start. So you presented when you were looking at the cut points for assays of kind of a bimodal distribution of negative versus positive, but is that really the case if you take a bunch of people in the general population? Do you really see clear bimodal? I mean, what I've seen from less experience is more of a sort of continuous distribution from very low signal to high signal. It makes me kind of just wonder about the the cut point question.
Of the cut point, right? So I think, I guess I can start with that, but I think it does depend on how you set up your assays and how you establish your cut point and what decile you use to establish that cut point. You can get a pretty clear distinction between positive and negative, but I think as you're pointing out, you'll have some overlap where the tails are kind of crossing. So there's some ambiguity there. I think in other assay formats, maybe you just see sort of a longer tail. It's not a perfect, bell-shaped curve. And it does get into this realm of, how do you determine, when you see this data, whether it's a binding antibody titer or whether it's a neutralizing antibody titer, what kind of information do you need to make a determination as to which patients are going to have efficacy versus which ones aren't, right? And I think that's kind of what we're all focusing in on at this meeting is how do we get there? What is there a way to get there? So.
I'm also just curious for anybody in the panel about tracking some of these maybe tab assays or even looking at the mass spec-based assays where you see patterns of transient levels of antibodies that are detectable and then they disappear, or probably some other proteins that might be binding, if you're talking about bridging assays, for example, where you're not looking at specific IgGs. Bridging assays could detect any sticky proteins that could be binding to the capsids. And so I was curious if that's a pattern that you've seen, or if we think there might be some opportunities to develop more longitudinal data sets in humans.
Actually, that is related to one question that I had. So, and Robert, you might be in the best position to answer this, I'm not sure, but do you think there have been enough studies looking at neutralizing antibody titers among individuals with low titers longitudinally? In other words, how much do those wander over time? And I think that could be clinically relevant for some people with low titer neutralizing antibodies if they fall below a certain level at some time. I mean, it might be difficult to deal with clinically because you'd have to be prepared to move right away, but how much do we really know about the stability of low neutralizing antibody titers in the adult and the pediatric population?
Yeah. That's a very important question. And I think, when we're talking about low neutralizing antibody titers, the assay matters too, because, what is the variability of your neutralizing antibody assay? So you do your qualification, validation of the assay, and most of the time, we do this twofold serial dilution, and then you determine that a subject is, 1 to 5 NAV. Well, the variability of the assay is a 1-2-fold serial dilution. What it means is that that patient could be 110 or in the limit of detection. Just give an example on that low range. So sometimes that variability is variability due to the assay. But nonetheless, there is a variability or your level of neutralizing antibodies that are influenced by the environment where we live. So your level of neutralizing antibodies against AAV will be different if you have the flu. They will rise or they will change. So how you are exposed to the environment will affect the level of neutralizing antibodies, especially when we're in this low range, like 1 to 5, 1 to 10. That's the natural immunity that we usually see. That 1 to 10, but it will be enough to exclude you from a clinical trial. Now, what is interesting was that chimpanzee study that is, very well characterized for 10 years. So there were low level of neutralizing antibodies that were very consistent in some of those chimpanzees. We don't have that in humans. We don't have that data set in humans. It would be nice to have. But sometimes you have that level of low neutralizing antibodies that not only in chimpanzees, I've seen it in monkeys, non-human primate, rhesus macaque and cynomolgus macaque. They stay low for a long period of time. And understanding why some and why not could be related to more virology, understanding AAV more. It's a commensal. We find it in tissues. That's how AAV was isolated, AAV8, AAV9 in monkeys, humans. How do we have actually replication once in a while? You get infected with adenovirus or with other viruses that revamp, that genome copies of natural AAV, and that expose, your neutralizing antibody rise because you have a memory response. So I think that's also not only immunology but virology. Question about AAV. What is the biology of AAV in humans?
And if we get re-exposed again to the wild type, right, AAV,
that it could be very different in monkeys. And I think, Juliette, you brought an important aspect, differences between humans and monkeys. And it happened with adenovirus, for example. Adenovirus, that it was the first generation of gene therapy vector, it has been shown that it's very, immunologically, it's very active in human and potentially dangerous. That's not the case in monkeys. There's more, you can work with adenovirus in monkeys. So.
Okay, Federico.
By the way, a while back we published data was a longitudinal study in humans where we did show that titers tend to be pretty stable over the years. This was a population of Duchenne patients, and the titers, even low titers, tend to be stable over time. But that's not my question. My question was, I was intrigued by some of the data that Juliette presented in monkeys, where you have, let's say, no titer or naive, let's say, for antibodies, monkeys, then you have like a low titer antibody monkey and a high titer, and you do see some kind of prevention of what looks like an acute liver toxicity in the seropositive animals, and the low titer, there's still transduction. Mm-hmm. So in a way, this is a good panel of people, right, to discuss, because, of course, there's that, but maybe my question is, what kind of toxicity would that prevent? A low titer antibody, by which mechanism? And then there's a whole other subset of potential issues related to complement activation and so on. So I would love to hear the panel and of course you Juliette, to comment on that.
Yeah, so this data set, it's really showing that the acute hepatocellular injury that happens within the first week, especially when the vector has a ubiquitous promoter, so again, that was a model with a ubiquitous promoter, seems to be directly driven by high level of transduction of the hepatocytes, and transduction being already active as soon as day 3. And we did some single nuclear RNA-seq, euthanizing monkeys that developed acute severe liver toxicity, and we had to euthanize them on day 3, day 5, day 6. And surprisingly, the single nuclear RNA-seq was always showing massive transduction and transcript at day 3, not only in hepatocytes, but in sinusoidal cells, Kupffer cells, B cells, T cells, all the cell types express transgene. Again, ubiquitous promoter, with the caveat that's a ubiquitous promoter. So it might be, I think, relevant to the Zolgensma type of acute transaminase elevation that they see, and somewhat managed to control with the use of steroids, but not always. There's some escape, and there's some patients developing liver failure. In these cases, maybe low level of antibodies would protect from that, from liver injury.
Hi, really fantastic session. Thank you all so much for the talks. I was wondering if you could comment on any relationship, if it exists, between pre-existing antibody responses and pre-existing T-cell responses. Because, and if there isn't a relationship, why not?
Well, I think it's a matter of detection. Most of the time, we can only use blood. And the good thing about antibodies is that they're secreted in blood, so we can detect them. The problem with T-cell immunology is with the T cells that you find in blood may not be the T cells that you're looking for. You know, you should go to the source of the T cells. It could be lymph nodes, the spleen, and actually, Kathy, hi, I mean, yes. Federico, you did a lot of work on this, and you know, a spleen is a good source of T cells, finding T cells and finding T cells against AAV. But I did a little bit of work on T cell responses after gene therapy, and actually, I did not find T cell responses in blood, but I found them in the liver. They were CD4, CD8, so they are there. The problem is they don't secrete. I mean, they secrete cytokines, but actually, you have to isolate those cells. So you have to go to the right place to find them. And in monkeys, yes, we can do that when we sack them. But in humans, it's more difficult. And I think, you know, Kathy, you can elaborate.
Yeah, I mean, this was a study that we did at Children's Hospital, and we collected spleens and blood samples from 44 patients who ranged in age from 2 to, I think the oldest one was in their 80s, who were undergoing splenectomy for non-malignant conditions. So they were basically healthy. They had hereditary spherocytosis or some other reason that they were, traumatic splenectomies. You know, they were involved in a car accident. And then compared T-cell responses and B-cell responses to AAV. And Robert is right that when you have, many, many more lymphocytes to screen, the incidence of positivity goes up pretty substantially. Comes closer to agreeing between B cells and T cells.
Yeah, maybe I can add to this that, so the study I showed briefly using just blood from healthy donors, that we've seen a perfect correlation actually between antibodies, so neutralizing antibodies and detection of AAV-specific T cells in ELISpot, but it was TNF-alpha ELISpot, which usually people don't measure. But the caveat of the study was that it was not hundreds of samples, it was around 20 samples. So I would say, it's not probably a very strong conclusion. But I'm saying that I agree that the assays here are critical, right? Detection sensitivity. On the other hand, I think it's possible that you have an exposure to a wild type AAV, and you would have antibody response without maybe having long-term T-cell responses, because if this, it depends, it's like vaccination as well, right? Some vaccinations have a T-cell component, some others don't, and it depends whether you have those damage signals, whether it's really maybe pathogenic virus or not. So I'm not sure in case of AAV, it may be also very person-specific, like on HLA type.
I think it's a really good point. It's always kind of struck me that there doesn't seem to be a very strong memory component to the T-cell response to AAV, whether that's pre-existing or even when it's post-dosing. I think post-dosing, when you look in the peripheral blood, you'll see that interferon gamma coming up as early as 2 weeks, but it's transient and it goes away in most of the patients, which is kind of curious when you're putting that high of a dose of capsid. You're getting the response, and certainly there has to be a CD4 component to it because you're getting class switching to high titer and high affinity IgG, but eventually you lose detection of that interferon gamma signal in the peripheral blood. And is that because you just don't have a strong memory response to this virus for whatever reason? Or is it because you're now looking in the wrong compartment? The cells traffic, they've gone back to the spleen or the lymph nodes, or they're in the parenchymal tissue where they're having their effector function, but they're not trafficking as much through the peripheral blood. So if you keep looking and you keep sampling in the peripheral blood, you may not see that same temporal correlation with some of the transient ALTs that have been seen and things like that. But I think, yeah, it's a really good question that deserves some more exploring, because even in the pre-existing condition, it's hard to make that correlation between antibody and T-cell responses.
Yeah. Thank you so much.
Thank you all for the wonderful talks. So my question is that can we use capitalize the inflammation management and cytokine skin storm management for seropositive patients?
I wasn't sure I heard the first question. Could you
Yeah,
repeat that?
inflammation management, like using non-steroid anti-inflammatory drugs for seropositive patients, and also using cytokine storm management, like anti-interleukin-6 and antibodies to management acute immune response for seropositive patients.
Can we use the steroids or IL-6 inhibition in seropositive patients, if I get this right?
In seropositive
Patients, yes. I don't know who has
do
an
you mean to try to
experience.
reduce the titer so you can treat them, or?
Want to management acute response. So positive,
So try to prevent the response
Prevent.
when you treat them.
Yeah.
So
So-
I think, oh, go
Go
ahead. That's just for IL-6. I think we have tested that in animals, and the problem there is that, so we had some response, so we basically could decrease the titers of antibodies in response to AAV. However, in the study we used low doses of AAV vector, and we didn't have any other toxicity, so we didn't have transaminase. So basically antibodies was the only readout that we could get from this study. The problem of blocking IL-6 pathway is also that it increases ALT. That's known side effects of drugs like tocilizumab. And then it's a little bit confusing, I think, to understand whether you see toxicity, hepatotoxicity due to AAV gene transfer, or is it due to the drug that you are applying, basically to prevent the transgene loss. I don't know if somebody would like to add.
Did you get lower titers?
Of antibodies?
Yes.
Yes. Yes.
Okay, so it worked for that.
For
And if your-
neutralizing
and if
antibody
your-
titers, yes.
If
Yeah.
your target for the transgene was not the liver, right? So maybe you don't care.
Maybe that's true, yes. Yes.
Thank you.
Just out of curiosity, I forget who showed the studies where IVIG was administered just a few hours after dose administration.
Yeah.
I'm curious, did anybody look to see whether that resulted in reduced titers?
Reduced titers to the AAV. You know, I'm not sure we did that in that study. We were just trying to figure out.
I'm just kind of wondering so you've put the capsid in, 2 to 4 hours later you neutralize it with antibody. Does that block it from interacting with the B cell receptor and you ultimately get a lower titer?
There's definitely at least one study in which immunization after it was intrathecal administration, then you have an systemic exposure. Whether or not that affects the downstream response, I don't know. Oh, thank you. So there's a study in which there is administration of, I think it was IVIG post intrathecal or something, administration of a vector, and so then you don't have peripheral exposure, but I don't remember if that actually mitigates. It was not one of our studies. It was a published study. I don't remember if that will knock down the downstream immune response.
That's a good question. I should go back and look and see if we did that. I don't know.
I guess I'm trying to think of the Rogan example, right?
Yeah,
where
yeah,
you give
yeah.
an anti-RH
Exactly.
to prevent.
Yeah.
But I
So-
think in some of the studies that we've done where we have administered virus and it hasn't been effective, it hasn't done what we want it to do, we still get antibodies. So I'm thinking of when we're trying to detarget the liver and we end up detargeting everywhere, we still get antibodies generated in response to that.
If you give it out.
If we give it IV,
Yeah.
yes, exactly.
Okay.
So,
Let me see, I don't know who was first, so. All right, go ahead.
thank you. So my question was around some topics that have been touched already with the BT interaction, but it has more to do with whether or not we have any insight on how long antigen stays on FDCs driving a germinal center response after an AAV infection. Because I'm not sure that we have a handle on that, and that could be relevant in terms of like how long ago a patient got sensitized and what is the current state of that immune response, as well as what happens when a patient gets dosed with their first dose of AAV.
So you're really asking about both situations. How long does the antigen persist in the setting of wild-type infection? How long does the antigen persist in the setting of administration of a product? And I would say that for the second question, we do have some data. I can remember a study that was done by one of your colleagues at Nantes, who I think was part of Horama, Juliette, that demonstrated persistence of AAV in the subretinal space for something like 4 years.
The capsid, yeah.
Yeah.
And in muscle as well.
So at least for product administration, I think it can persist for a very long time, and maybe that's why those titers never ever go down. I don't know. I don't know if that's been looked at as carefully for other routes of administration, but that was in, I think, non-human primates, if I remember right.
Similar data in muscle. 4 years later, I still have capsid in muscle.
Yeah. And in terms of wild-type infection, I don't know. I don't know if people, I mean, it's usually through the respiratory tract. I don't know how long it persists.
Probably two very different problems, though, because in a wild-type infection, soon after exposure, before there's significant replication, there are antibodies formed that begin to clear the material, and you really never develop full-blown systemic viremia like you do in a clinical setting with a therapeutic dose. It's massively higher amount of capsid. And to your question about muscle, to Juliette's point, I guess that's primate data, but we did a study in alpha-1 antitrypsin vectors given IM, and their capsids are present in the muscle 1 year after exposure, and presumably longer than that. They're incredibly stable.
Do you know where they are in the muscle?
Interstitial spaces. So my question, though, is about assays. So there was a lot discussed about the methods we're using to detect antibodies and their significance. And we're using the same methods to detect pre-existing antibodies as we are post-exposure. But I think in general, the pre-existing antibodies are low affinity and non-specific, and the post-exposure antibodies, which are also polyclonal, but they're much higher affinity and very specific. So can you comment on that and whether we should be developing other strategies like an antigen-specific inhibition component of a TAB assay or other methods you think might be relevant, including B-cell clonal sequencing, for example?
Oh, yeah, that's a good question. It's a very good point, though, right? And I think, Kathy, you brought it up early on, too, that the titers that you see prior to dose administration versus after are two very different questions. After dose administration, you have very fine specificity for the capsid. That was the input capsid of the gene therapy vector. It will have some cross-reactivity with other vectors as well, and I think some of the maybe some of the lack of specificity you see in the pre-existing condition is that you were maybe exposed to a different AAV capsid than what you're actually measuring. AAV2 and 8 are much more prevalent than 5, for instance.
It's cross-reactive.
And it's probably cross-reactive. When you're looking at AAV5 in particular, a lot of what you're actually measuring may be cross-reactivity from AAV2 or AAV8. AAV5 is a little bit unique in that it's not a primate isolate. It's more related to a goat isolate, I think. So there's some questions to really how much that particular capsid actually circulates within the human population. So yeah, beyond that, I'm not sure how to answer your question. Like, what else should we be looking at? Yeah, B-cell receptor sequencing could be one way to go, and maybe that's something that's a little bit more precise in its quantitation, I guess is one way to say it. But then I still think you're going to be stuck with this question of what do you do with that information? You've gathered it, you've detected it. How do you know which patients are still going to be the ones you can dose versus the ones you probably shouldn't? Right?
I mean, I think it's relevant too to the point I'll make in the later session that none of the assays we use actually differentiate seronegative status from truly naive until there's an exposure. And then when you assay IgM, you can differentiate those two conditions because they look the same on either the NAB or the TAB assay. And I think I think it was alluded to that those, in your study, Juliette, the animals that were seronegative, if they were in fact naive, they have more predominant IgM response and probably more acute injury. At least we've seen that clinically in patients who had cross-reactive antibodies or were not truly naive and had a predominantly IgG response. It's slightly shifted to later. It's not within the first 5 days. It's more after day 7. And that can be even a higher magnitude effect through the very high IgG1 that comes from the memory response. So it's a conundrum that I don't know exactly how to deal with yet. But I think it's because it will be really helpful to be predictive about those patients who are more likely to have acute systemic inflammatory responses. So you can be prepared to counteract those as opposed to patients who may have there's just such wide variability in immune response profile. And
when you say sort of truly naive versus seronegative, for the naive population, are you thinking of people that haven't been exposed to any AAV capsid at all? So there's no, no cross-reactivity.
So I'm going to ask one other question, which is, Brian, you looked at all your data and decided to use TAB for screening. And Sharon, you looked at all of yours and decided to use NAB for screening. Did I understand that right? No. Which were you doing?
So we have used both here in the preclinical space. For our clinical, we have not disclosed which assays.
Okay.
Yeah, but the data I talked about was mostly TAB. We just saw a really nice correlation in the data set that the cohort that we were evaluating between the two.
So then my question was this. You saw a really clear correlation, and Brian had multiple instances of patients who were positive in one and negative in the other. And do you think that that's a difference between pediatric and adult subjects, or do you think it reflects a difference in how you do the assays, or what do you think is the explanation there?
I think in our case it's an issue of assay sensitivity. So you can make these neutralization assays very sensitive on a plate in vitro, and at that lower end where you're not able to detect any antibody that correlates with that neutralizing titer, I think it doesn't have any effect in vivo. And so from that perspective, and people are free to disagree, it's kind of a controversial topic, right?
It's a point of the discussion, right?
In that case study, let's say the TAB assay was the right one that was able to predict who was going to have efficacy versus who was not. Now I say that because it's a limited data set of people that actually turned out to have a titer, right? There are those 3 patients that were negative at screening and positive at day 1. And it kind of gets back to that earlier question about what's going on with these really low titers that are just kind of hovering around the cut point. I think, as you were alluding to, different inflammatory states might boost that pre-existing antibody titer at different times. Nonetheless, what we had showed, those 3 patients were kind of in the lowest quartile of expression values, whereas the 11 patients that had a neutralizing titer at day 1, and again, we didn't screen for that in that study, their efficacy values were actually a little bit above the mean for the ITT group as a whole. And so that, I want to be careful here because I think a lot of this has to be empirically determined for each and every gene therapy product, right? There's a lot of desire to say, well, it's AAV5 and it's made in an insect cell, so it's going to behave the same, right? But the doses are different, the transgene is different, the promoters are different, poly-A tails are different. These all behave differently once they get into the nucleus, and that probably is the reason why we're using different doses in these studies as well when you're comparing Heme A and Heme B, again in that example. So although I totally understand the desire to say it's AAV5 or it's AAV2 or it's AAV9, this is the titer in this assay above which you can dose and which you can't. There are other factors involved, and I think that's kind of the difficulty, is pulling all of that diversity out and trying to say what's the
Okay, we have 2 more questions here, and then we're going to break for lunch.
All right. So following up on what Kathy said in terms of this discordant, NAB versus TAB, I think we all have seen that there are neutralizing factors that are other than antibodies, and I think we understand and have characterized that. But I've always been curious about, what do we know about binding antibodies that don't neutralize? And should we be excluding individuals, as Roberto just showed, individuals that have binding antibodies that don't neutralize, especially given what Klaudia showed as well in terms of AAV5, where you may have these binding antibodies that don't neutralize, and people may still have a benefit. I feel like as a field we're moving to TAB in part because it's a simpler assay, easier to standardize, easier for regulatory, and I understand all of those complications, but I wonder from anybody on the panel, what really is their thoughts about, should we really be excluding individuals, or what are the function of binding antibodies that don't neutralize AAV?
And to add, so they can even enhance the transduction, right?
Sure.
That
They
has
do.
been shown as well. So
Yeah.
that's complex.
Yeah, I just following on that comment, I also had a thought about that, which is that, if I recall correctly from your talk, Brian, in those assays you're starting with a capsid coat, and then you're floating in the sample. And so I think that intrinsically you have like natural competition of IgM and IgG happening there that might impact what you're detecting with your detection antibody. If you start with a coding for either IgG or IgM, and then sandwich your ELISA in that way, you might get a better quantitation of what IgGs and IgMs are there that are actually binding, and if we believe that IgGs tend to be the more neutralizing ones, that could give you a better handle on things.
Yeah, it's a really good point. There's multiple flavors of these assays and how you can set them up, and I think every single one is going to give you a slightly different result. And you can set up multiple assays where you're separately detecting IgG and separately detecting IgM, and certainly that's been done. And when that's been looked at across a set of samples, you'll find that even where you're detecting pre-existing immunity, it's not only polyclonal, but it's a different mix. Some people have a lot of IgG, others have a pretty good mix of IgG, IgM, even IgA can be detected, and so I think that's where some of this variability is coming from as well. And it brings up this kind of issue, maybe this is just my opinion, that I've always had this idea of a titer threshold. Let's say you've determined in some non-clinical study that it's a titer of 100. I think the trouble with that is that there are multiple ways to get to a titer of 100, right? You could have a very small amount of high affinity IgG, or you could have a bunch of low affinity IgM, and those are going to have different effects in vivo with respect to neutralizing or causing off-target effects of the capsid.
Yeah, and depending on how you set up the assay and how you run it, that titer is always going to change. So it has to be a consistent assay.
There is guidance on how to establish the cut point of the assay, and that's very useful for saying you're positive or you're negative, right? It's then what do you do with that information, and do you titer it out further? What dilution step do you use? And then again, what's the composition of that antibody response that got you to that titer? It's not always going to be the same. And I think that's why you're seeing some of this variability in the clinic, and that one patient with a titer of 50 is fine, and another one has no efficacy whatsoever, right? So,
okay, I think we're going to have to wrap it up, and I want to thank the speakers and the audience for highlighting all the things that we still don't understand very well. And I look forward to the afternoon session. So we need to be back here, Michelle, at 1:20. All right, it's going to be a quick lunch. Okay. Thank you.